Good morning, good afternoon, good evening, wherever you are. Welcome to this first of our 2024 translational seminar series. These series are a joint initiative between the International Cholangiocarcinoma Research Network, the ICRN, the ENS-CCA, European Network for the Study of Cholangiocarcinoma, and the Precision BTC Network, which is a cost action based in Europe. I'm Juan Valle. I'm the Chief Medical Officer of the Cholangiocarcinoma Foundation, and together with my co-chair, Professor Chiara Braconi, who is Chair in Hepatobiliary Oncology in the School of Cancer Sciences in Glasgow, and she's also the Vice Chair of the Precision BTC Network, we'd like to introduce you. We'd like to welcome you to the seminar series. Before I hand over to Professor Braconi, just to go through the introduction to our speakers, a few practical points for the session ahead. So we would invite people to keep their cameras on so that it allows for better interaction. There will be attendance certificates for live attendees, so we will send those out after the meeting. We would like to encourage open and spoken questions, but if you want to use the Q&A as an alternative, please do so. But it would be great to have an interactive session where people feel free to jump in, ask a question. Both the speakers are quite happy to be interrupted if necessary. At the end of the meeting, we will ask for your feedback, and part of that will also include maybe some suggestions for some future meetings, so that we can then have a number of seminars over the year. So without further ado, I'll hand over to Chiara, who will introduce the speakers. Thank you, Chiara.
Thank you, Juan, and good morning, good evening, everyone. Today, it is our first series, as the first seminar, as Juan said, and we have the privilege to host the two great speakers who will be talking about KRAS as a therapeutic target in biliary tract cancers. So the first speaker will be Thomas Karasic. He's an assistant professor at the University of Pennsylvania, where he is also physician lead for the development of therapeutics in the hemato-oncology division. And then it will be followed by Jarman, who is a Post-doctoral Research Fellow in the MRC Human Genetics Unit at the University of Edinburgh in UK. So Thomas, we can start with you, and it will talk about targeting KRAS more from a clinical perspective. Thank you.
All right, thanks, Chiara. Let me share my screen. So I am a medical oncologist, and so I am very excited to get to present some of the new data, the new clinical data that's emerging from these KRAS inhibitors as they begin to show some early signs of efficacy in the clinical space. So I think, as many already know, molecular drivers vary quite a bit in cholangiocarcinoma depending on the subtype. Most of the development of the molecular therapies that are available, FGFR inhibitors and IDH, have been focused largely in intrahepatic cholangiocarcinoma, where most of these mutations are seen. But in perihilar and extrahepatic cholangiocarcinoma, HER2 amplification and KRAS mutations are the more common molecular targets. And this is shown in the graph below, where you see, again, 30 to 35% of these patients have KRAS mutations as a molecular driver. So the KRAS mutations really are the largest unmet need of molecular therapy in this disease. And of these KRAS mutations, G12D is the most common in cholangiocarcinoma. The numbers vary a bit depending on which series you look at. This is a series on the left, the pie chart, out of 550 cholangiocarcinomas of all different subtypes. And again, about a third are G12D. And then the graph on the right is from resected intrahepatic cholangiocarcinoma patients, 1,000 of them. And again, G12D is the most common of the KRAS mutations in those patients. And there are other series as well that show the same thing. I think it's also been shown over a number of different studies that KRAS mutant cholangiocarcinoma seems to have a worse prognosis than wild type. The graph on the left, the Kaplan-Meier curves, this comes from that same intrahepatic series of 1,000 patients with resected intrahepatic cholangiocarcinoma. And again, the wild type KRAS mutation patients seem to do better than those who had a G12 mutation, G12D, G12V, or other G12 mutations. Interestingly, non-G12 mutations didn't seem to have a worse prognosis in this series, although the numbers are relatively small. And again, this has been shown in other series, largely from resected intrahepatic cholangios. The graph in the top right is another one of those series, and there's been a meta-analysis. And then the graph on the bottom right is from a perihilar series, which again showed the same thing, that KRAS mutant resected perihilar does worse than KRAS wild type.
Tom, before we move ahead, can I just bring you back to this slide and ask you a question? I mean, you've shown that different variants of G12 is associated with different prognosis. Do we know if it is related to the variant per se or to some other co-molecular alterations that occur during the carcinogenesis?
I don't know that we know completely, because a lot of these are fairly limited molecular panels from older series of resected tumors that largely looked at just KRAS. In the series from Princess Margaret that I showed the graph of with the KRAS alterations in the first one, there didn't seem to be any worse prognosis with the other molecular drivers that seem to come along, or the other molecular alterations like SMAD4, P53, or CDKN2A. That didn't seem to be prognostic. It seemed to be KRAS. But I don't think we know. And we also don't know that well in advanced disease because we don't always have outcome data and molecular data in these. These are largely, again, from resection series.
And Ed, would you agree with that? I mean, and probably you will discuss some of the data later. Would you say that KRAS on its own is enough to drive the disease, or do we need something else?
Well, what we see in models is that KRAS isn't sufficient on its own, KRS G12D. If you just have that, then typically cells will senesce or get cleared. In the models that we use, typically you need something else, something to protect those cells from being cleared. And the most common kind of culprit for that is P53. But if you look at the frequency of mutations in patients, P53s doesn't always co-occur with KRAS. There's plenty of patients that have just one. So my suspicion from the modeling is that you do need other pathways to be perturbed to protect these cells from the kind of negative effects of KRAS oncogenes. But whether that's through alternative mutations to the P53 pathway or alternative pathways that protect cells from senescence isn't entirely clear. But I do suspect there's a combination thing going on. And I think what I'm going to talk about, in fact, is the rarer mutations and whether they might kind of collaborate to facilitate the actions of KRAS. But I definitely don't think KRAS alone is considered sufficient, if that answers the question.
So if it is sufficient for initiating the tumor, maybe we don't know yet, but definitely from the data that Tom is presenting, it looks like there is an association with prognosis and tumor development. So still there is a rationale to target KRAS, right, Tom? So maybe worth it to see, what next is coming. Please continue.
Maybe if I can just quickly jump in also, Tom, do you know if this KRAS is an early event like we see in pancreas cancer and some other cancers? Do you know any of that from the clinical data, or maybe Ed will cover that later on?
I was going to say, Ed is probably better equipped to answer that question. I mean, there are, again, there are fewer models of cholangiocarcinoma genesis, and so I don't know if we know. But as he said, these models generally require KRAS plus something else. So I don't know if we know. Ed, you may have better sense.
So in a modeling context, KRAS alongside something like P53 is sufficient to initiate. Whether that relates to patients having these mutations early at the start is unclear. When we model it in mice, we can cause cholangio through just two mutations, KRAS, P53, for example, but it doesn't necessarily mean that in the patient context, these are the first mutations. I suspect they might be, but I don't think there's much evidence either way at this point. It's hard to kind of pick that apart. Obviously, patient data, you've got late stage cholangio by that point. So, but in the modeling context, for sure, it can be an early event, a triggering event, not on its own, but with just one other alteration is sufficient.
Yeah, I don't think we have the same data like in colon cancer polyps or in pannins or other things to sort of see. I mean, maybe such data, could be looked at, but I don't know that it has been to see as KRAS common at the beginning.
Thank you, Tom.
So getting
from Bridgewater, if you don't mind, before moving ahead, and he asks if there is a pattern simply looking at these common mutation that would differentiate the cholangiocarcinoma, KRAS cancers from other like pancreatic cancer. So, Tom, according to the profile, would they be different in the type of mutations and pattern of mutations, or are they very similar to KRAS mutant of any other type?
I mean, often they do have many of the same molecular alterations as is seen in pancreas cancer. Again, the common ones, P53, SMAD4, CDKN2A, as are seen in pancreas cancer, are also often seen in KRAS mutant cholangiocarcinoma, at different rates. And again, there are other molecular alterations that I think are seen a little bit more in cholangiocarcinomas, co-alterations with KRAS, so PIK3CA and other things, but certainly they do tend to resemble more genetically pancreas cancer than other forms of cholangiocarcinoma.
Okay, thank you. Please carry on.
All right, so I will now get to talk about some of the exciting clinical data we've started to see with these KRAS inhibitors as they've emerged. And our data in cholangiocarcinoma is still fairly limited, but I think so far has suggested that KRAS is a molecular target, at least G12C. G12C is fairly rare in cholangiocarcinoma. It's about 1%. And so this is not likely to be a huge population of patients who benefit, but it seems like so far from the drugs that have presented data that it is an effective target. This is data from adagrasib. This was published in JCO last year, and there were 8 patients with cholangiocarcinoma in this series. And as you see, all of them had disease control, 50% had objective responses, and the median PFS in these 8 patients was 11.3 months, and median OS was 15 months. And this is in a refractory cholangiocarcinoma patient population. So really impressive results, although a limited sample. And then a couple of weeks ago at GI ASCO, we saw another KRAS inhibitor, gleserasib, which again showed impressive efficacy in G12C mutant biliary tract cancers, 5 out of 7 with objective responses, and again, 100% disease control rate. So it again suggests that this pathway is targetable, or at least G12C is so far. But as I had said, it's relatively rare to have G12C, so it's unlikely to move the needle significantly. I think the other KRAS inhibitors that are beginning to emerge are a lot more exciting. And I think it's been fairly clear with recent data that G12D is also a druggable target. G12C was the first because it's possible to make a covalent bond in the switch II binding pocket, which is where all the available G12C inhibitors bind. They form a covalent bond with the cysteine there. But with, I think, impressive medicinal chemistry, new drugs have emerged that also can target that pocket. MRTX 1113, this is a graphic shown at the bottom, can also target that same pocket and bind KRAS G12D in the inactive GDP-bound state. And then there's been other drugs, and again, I've just given a sampling of some of the names that are out there. There's far more than this that are entering, that are in preclinical and clinical testing. But the other one I think that's most interesting because it's started to show data is the RMC series of drugs. And these use a molecular chaperone, cyclophilin A, the same thing that cyclosporin binds to, and makes a binary complex that then binds to the RAS in the on state, and then makes an inhibitory tricomplex. And again, there are the RMC compounds. There's a G12D specific one, which is in testing but hasn't yet presented data, and then a multi-KRAS, which I'll show data from in the next couple of slides. And then there's a degrader of KRAS G12D, which is also an interesting approach, ASB 3082. And so again, as these in the next couple of years, I think we're likely to see a lot of these drugs begin to present data. It'll be very, very interesting, I think, to see how these drugs behave, what the toxicity profile is. But right now, it's very early days. And as you see, I think from the PDX models, again, the one yellow bar is the one cholangiocarcinoma model in there. Again, it sort of speaks to what we tend to see here, 1 or 2 cholangiocarcinoma patients in these studies as well, and really a need for us to get more cholangiocarcinoma patients on these studies. This, though, is the data that came out at ESMO from the RMC 6236 molecule. Again, this is a multi-G12 KRAS inhibitor. that, again, makes the inhibitory tri-complex in the on state. And this is data from expansions in pancreas cancer and non-small cell lung cancer. And as you see, very impressive waterfall plots. Response rate was a shade under 40% in the combined population, and disease control rate, again, above 90%. Again, most patients seem to derive some disease control from these, and this is in a refractory pancreas and non-small cell lung population, so very impressive. And again, this was all different G12 mutations, D, V, and R being the common ones, A as well in the non-small cell lung cancer patients. And it seemed to work regardless of the G12 mutation.
Tom, so some of these response rates do seem quite impressive. Is there anything you can say around the durability of the responses, and does it vary across different tumor types?
I don't think we know yet. I don't know that, I don't actually recall how many even were far enough out for that had progressed at the time they presented the data at ESMO. I think, again, they keep updating the N and showing waterfall plots. And there are some swimmer plots, and some of these do seem to be reasonably durable, but the swimmer plots have a lot of different dose levels, some of which are probably going to be below, you know, what's looked at in phase 2. So I don't think we know the durability quite yet. Again, at least from the G12C experience, it seems to be vary quite a bit among diseases as well. You know, more durable in some and less durable in others. So I think we'll probably see that over the next few months as, you know, as these data mature a bit more.
Thank you.
And Tom, can I ask you another question? In the waterfall plot you have shown, I think that we've seen some very good responses independently on the dose. So it seems like some lower doses can give some very good responses. Can we speculate why that happens? Is probably is not dependent on how much they interact with the targets or any other justification?
I mean, I think it just speaks to the, you know, again, the potency, at least of this drug, that it does seem to have responses even at much lower doses than are tolerable. And I think it supports, again, efforts, you know, from the FDA and other agencies to look, you know, with the dose optimization, so that we aren't just treating all patients at the maximal tolerated dose. I think it's certainly possible that the best dose for these drugs may be lower than the maximum tolerated, because again, we are seeing responses at much lower doses.
Mm-hmm. So just a good selection of the patients is the key point here.
Yep. And this is, again, the toxicity that was seen with this, and the primary toxicity of this drug is skin. Again, more than 80% of patients had some type of rash, and that was the main reason for dose reduction. About 15% of patients required dose reduction due to rash. Again, these were typical of MAP kinase inhibitors, the types of rashes: acneiform, pustular, maculopapular type rashes. GI toxicity, also very common: nausea, vomiting, and diarrhea, stomatitis, and fatigue. I think importantly, though, you know, as with that, we didn't see a lot of organ toxicity as has been seen in other drugs that have targeted the MAP kinase pathway unsuccessfully and RAS unsuccessfully in the past. There is not a lot of liver toxicity here. There was no report of cardiac or ocular toxicity or lung toxicity. So again, while these were fairly high rates of toxicity, there didn't seem to be the more serious toxicities that have been seen with other drugs in the past.
And would you say that the toxicity profile is different between the PAN inhibitor versus the G12D or C so far?
I mean, we don't know yet for G12D. I think it'll be very interesting to see as that data emerges. You know, is the RAS or is the rash driven by G12D, or is it something else? Because we don't see a lot of rash with the G12C inhibitors. We see the GI toxicity, the nausea, vomiting, and diarrhea, but we don't see nearly as much rash. So it will be interesting to see if that is driven, you know, by targeting G12D, or if some of the G12D specific ones don't have that same toxicity. And then there's going to be drug-specific toxicities, again, different rates, I think, of off-target effects, you know, liver toxicity, other organ toxicity. So again, that'll be important as we start to see more data emerge, you know, which seems to be the best. Because unfortunately, as these drugs tend to develop, we often are left with a bunch of non-comparative studies with efficacy data, and we're often choosing based on toxicity profiles. I think it's also important as we start to think about these drugs as they emerge that resistance is inevitable with these molecular targeted therapies. These are not curative therapies. And I think we can learn a lot from the G12C story where, you know, as expected, there are a number of resistance mechanisms, upstream signaling from receptor tyrosine kinases or amplification of upstream signals, as well as mutations in downstream pathways like BRAF or MEK. Other mutations within KRAS that either prevent drug binding or cause another activating mutation, or activation of parallel pathways, NRAS or PIK3CA, or mutations in SHP2 or alterations in that pathway to, again, prevent the drug from getting into the inactive state where the drug binds. So again, it's likely we'll see a lot of these same things emerge. Some of them will be drug specific. For example, a G12X inhibitor, probably G12V or R won't be a big resistance mechanism, but a G12D specific one, you know, that may be more of an issue. So I think, again, there's already plans, obviously, to look at resistance in patients in these studies. And it's important to recognize that some of that may vary from disease to disease. So we really do need cholangiocarcinoma specific data here. Combos, I think, are going to be key. And I think probably the most exciting combo, again, because these are not going to likely to be curative therapies, they're going to be palliative, and resistance will emerge, is that there does seem to be quite an interaction with the immune system with these drugs. This is data from pancreas cancer from 2 different labs, looking again at the G12D specific drug, MRTX 1133, showing M1 polarization, increase in cytotoxic T cell infiltration, increased fast ligand in the tumor cells. And this is a mouse experiment showing that if you take away CD8 T cells, you really lose the effect of the drug. The black is the vehicle, the red in the middle graph is with an anti-CD8, and really you see there's very little effect of the drug if you take away the T cells there. Taking away B cells doesn't seem to have a difference. The blue and the dotted lines, that's drug with or without a CD19. But then when you add a CTLA-4, you really see, again, a increase in terms of the potency with or without a PD-1 inhibitor. So again, I think that is very exciting to think that we'll be able to use these to increase the immune response. And it's certainly something that needs to be looked at early on in combination studies. And then I will just end again with a few, I think, takeaway points. Again, KRAS is very common in cholangiocarcinoma. I think the drugs that are emerging have the potential to really impact a large number of patients with this disease. G12D is the most common, and so the most important for us as drugs are being developed. And these drugs are looking both tolerable and potent in other diseases. We really need to see data from cholangiocarcinoma in these studies, not just N of 1 or N of a couple. We really do need cholangiocarcinoma specific cohorts in these studies. And then I think most important is if these drugs, you know, emerge, we shouldn't wait years for them to find their place in advanced disease before we start to think about how can we use molecular therapies, including KRAS inhibitors, in the curative setting, in the neoadjuvant and adjuvant setting, and start to think of how to design umbrella type studies where we use some of our molecular therapies in curable disease. So with that, I'll end and take any questions and turn it over to Ed.
Thank you, Tom. Just a couple of things to pick up on. Obviously, we're seeing some activity in monotherapy, and you've talked about kind of 2 approaches going forward, using it in combination with other pathway inhibitors to see if we can overcome some of the emergent resistance, or combining it with immunotherapy. In terms of drug development, where do you think this is taking us as kind of the next steps? And are we going to, do you think, be starting off with monotherapy and then using salvage treatment on progression, or are we going to be going down the combinatorial approach? And is it with other targets or with IO?
I think, again, the IO, you know, is more exciting because of the potential for long-term disease control if we can get an immunotherapy response. I think with targeted therapies, what we see with other molecular targeted therapies is that resistance tends to be multiclonal. It is often not just a single resistance pathway that develops. When we do liquid biopsies in these patients, we often see multiple different pathways emerging all at the same time. And so I think that adding molecular combinations at that point may not be that effective. I think we need to be thinking about what's likely to emerge and target those potentially upfront in combination. You know, again, that will also add toxicity. But I think, especially if we're doing it in earlier stage diseases, that it would be really, I think, important to think of combinations from the get-go.
Thank you, Tom.
Thank you.
And you have alluded already that probably there are different mechanisms of action in different tumor types. So we have learned the lessons from the G12C inhibitor that, the brilliant results we've seen in lung cancer not necessarily reflected into the bowel cancer. So I agree that probably we really need to have the evidence in biliary tract cancer before we can we can introduce them into the clinic. So with this in mind, I think the talk by Jarman would probably be insightful, and he will talk about the cholangiocarcinoma mutation heterogeneity and modeling genetic interaction with KRAS G12D. Thank you. Ed.
Thanks. I'll just share my screen. And you can see that okay?
Yes, good.
Lovely. Okay. Yeah, so what I wanted to talk about today is I guess the broader mutational context of cholangiocarcinoma with KRAS G12D mutations, and how we can model this heterogeneity and try and pick apart how this wider context might be affecting disease progression and tumor biology. So I'm a postdoctoral research fellow in Luke Boulter's lab in Edinburgh. So we do a lot of kind of preclinical mouse modeling and things like that. That's really our expertise. Really, this boils down to this issue of, as I said, cholangiocarcinoma is mutationally heterogeneous. These kind of blue bars at the bottom of the slide represent mutation frequency of a large panel of mutations detected in patients with CCA. And as I'm sure most of you know, you get these really common mutations at the left-hand side that are relatively high frequency. I mean, even the most common P53 is up to 30-ish, maybe just a bit above that percent of patients. So even the high frequency drivers aren't hugely high frequency. The issue that we're interested in is this really, really long tail of rarer mutations. And we want to ask the question, do these impact the biology and the action of the more common driver mutations? So the more common driver mutations, including KRAS, are the most attractive targets because of their higher frequency, but the efficacy of targeting them isn't always nailed on, and there are obviously resistance mechanisms, but we're interested in the question of, are there any pathways driven by co-mutations or rare mutations that might modify the behavior of tumor cells with, say, KRAS mutations and affect patients' response to KRAS targeting, for example. So it's this question of, do rare mutations interact with these more common drivers? And this is also kind of includes the problem of a lot of patients don't have detectable driver mutations, or these more common detectable driver mutations. So do rarer mutations work together to activate these pathways in different ways? So it's a complex question. It's something we've been trying to model. And as a little sidebar, I just wanted to mention mutational heterogeneity based on disease etiology as well. So it's been mentioned by Tom that different anatomical locations, intrahepatic cholangio and extrahepatic, have different mutational frequencies, and that's hugely important in how we think about treatment. The same can be said in this example about fluke versus non-fluke associated cholangio. But the point I'm more trying to make is that these studies that look at mutational frequency are just about able to pick up differences in these high frequency mutations, and we're just about getting to a point where we can confidently say that these differences are there and quantify them, still with quite a level of discrepancy between studies, but to some extent we can see the difference. When it comes to rarer mutations, there's just not statistical power or the numbers, so it's really hard to know whether effects from rarer mutations might be different between disease etiologies. It's another kind of complexity to the question. So I'll point out, for example, that between fluke-free and fluke-driven cholangio, in this study there was no significant difference seen in KRAS mutational frequency, but that doesn't mean that these don't have different mutational backgrounds that are modifying KRAS behavior in these tumors. So without being kind of too negative about it.
Can I just stop you there and ask you a question? Because Related to what you're alluding, that there might be, you know, some background that affects the KRAS mutation driven carcinogenesis. In the previous slide, you mentioned that 30% of cases are still without a known genetic driver. Do you think because we have not, we don't have the tools to identify those rarest mutations, or do you think that there are some other backgrounds, even non-genetic backgrounds, that we need to consider, and sometimes it might be in some specific cases more important than the genetic?
Yes, I think it's a kind of combination of two things. I think there is a kind of study strength and an issue with numbers with cholangio, which is always one of the issues, where if we were able to get far more data, far more patients and full exome sequencing, then some of these, what we're calling rare mutations that are hard to define, might become more defined. And it might be clear in some patients which don't have clear drivers what those drivers then are. But actually, in more context, I'd suspect that we need to look or think at the pathway level rather than the mutation level. And so there'll be many different ways to activate specific pathways. And in cholangio, with such a broad range of mutations, different patients might be have their tumors working through the same pathways, but through different kind of mutational origins. And I suspect that might be part of it. But until we have deeper and more complete exome sequencing across much larger cohorts, it's a little bit difficult to tell in some ways.
Yeah, Ed, we're also dealing here with kind of heterogeneous, group of patients, all called cholangiocarcinoma, with differences in intrahepatic, perihilar, and distal. But I guess there are also, in addition to the differences that you've seen with liver fluke, yes or no, we know that there is a group of patients where there is a very strong inflammatory driver, the PSC related to ulcerative colitis and Crohn's. Do we know there's any difference in KRAS activation in the inflammatory group, if we want to call them that?
So, I mean, I think it's still quite hard to even define them as a singular inflammatory group. But in terms of PSC to cholangio, there is a decent number of KRAS mutations detected in that cohort. And once again, kind of P53 as well is very high in PSC to cholangio. So there seems to be, in my mind at least, an interplay between KRAS and P53 in a way where the PSC inflammatory context maybe matches mutationally to some extent the fluke inflammatory context. I think there is a clear difference in inflammatory drivers and how they're promoting different kind of mutational stresses. And I guess I get the impression that there will be some way to stratify those, but I don't think it's quite clear yet. I think understanding a little bit more about the mutational mechanisms and how exactly those inflammatory drivers are creating mutations might be informative. But at this stage, I'm not entirely sure we can categorize them cleanly, if that makes sense.
Thank you.
Other than to say they're higher frequency, KRAS is seen in PSC. I can't really comment on other inflammatory causes. Actually, I don't really know the mutation rates in those contexts, but there are some anecdotal examples that different inflammatory causes correlate in terms of mutational frequencies, if that makes sense.
Thank you.
So yeah, I mean, I think one of the points I wanted to make here was that there is maybe a need for a little bit more data on this in terms of even in terms of frequencies, and not just in these studies looking at the core drivers, but trying to get whole exome sequencing and understand mutational rates across the board, so that we can maybe pick out some of the frequencies of these rare mutations and understand what the commonalities might be. But we're not quite there yet. So that was a little bit of a sidebar in some ways. What I want to talk about is our work in the lab looking at interactions with KRAS. So looking at these rarer mutations and understanding how they interact with that mutation, KRAS G12D. And this is the output of a paper by 2 ex-PhD students in the lab, Nick Younger and Molly Wilson, published mid-2022. And the aim here, as I say, was to look at these rare mutations and see how they interact. So the first kind of barrier to this and the difficulty that you've got to approach is how you define your rare driver mutations, because we compiled exome sequencing from 6 different studies and found a total of 55,000 mutations within those data sets, and you have to find a sensible way to compile them into what we perceive as potential drivers. So our approach to this was to use a pipeline called Intergen. And what we've done is we've focused on mutational frequency, specific hotspots for mutations, and mutations that indicate a functional effect, so in a catalytic domain or something like that. And using this pipeline, we've been able to whittle down, as I say, 55,000 mutations from these data sets, from these exome data sets, into a set of what we perceive to be driver mutations from cholangio patients. So this is a patient-led screen, and these genes have all been defined from patient mutations. Within that set, what we see is our kind of usual suspects, our canonical driver mutations, including KRAS. No surprise there, and these come up at the highest frequency for sure. We identify a set of drivers that are in cosmics, are recognized on a wider scale as being drivers in cancer. And then we have a set of 60 or so novel driver mutations. So there's a whole plethora of rarer mutations that are predicted to be active or predicted to be drivers in some way, that are present in cholangio patients, but have no indication in the literature really that they're recognized as current drivers of cholangio. So this set of 100 or so mutations acted as a pretty sound basis to screen for interactions with KRAS mutations. And the approach we took in terms of models to do this was this hydrodynamic injection model. So what we can do is we can inject naked DNA plasmids into the mouse tail vein at high pressure, and these plasmids get taken up by hepatocytes and expressed. So we can do this with CRISPR-Cas9 machinery and transposases to ensure integration into the genetic material. And we can end up with situations where whatever library of guides we want can be expressed within hepatocytes in the liver 24 hours after injection. This example here is just to show GFP expression after modulation with a LOXP guide, but this is just an exemplar of the model that we can target these hepatocytes with CRISPR-Cas9 machinery. What we do in our actual screen is include our transposase, which drives integration of these genes, as well as a library of these 100 or so, or 96 driver mutations that we've identified and whittled down with the Cas9 machinery and KRAS G12D, or in this study NRAS as well, as our oncogenic driver. So we're going to combine our oncogenic RAS drivers with our entire library of novel drivers and see which ones are sufficient to drive cholangiocarcinoma formation. And so this was the setup for this study, and this is how we approached it. It was so after injection, if you just put in, to answer kind of the question from earlier again, if you just put in KRAS G12D or NRAS, which we did additionally in the study, you don't get tumor formation. This gets cleared, the cells tend to become senescent and it gets cleared by the immune system. So you need some additional driver on top of KRAS to get tumors. When we add our library of guides, 3 guides for each of the 96 mutations, we suddenly get formation of cholangiocarcinomas after 8 to 10 weeks in these mice, and we get them at fairly decent frequencies. So the next question becomes, what are the guides that have induced mutations in these tumors? So we performed exome sequencing on a number of tumors from these mice and identified which indel or deletions occurred close to these guide targeting sites within these driver genes of interest. Not surprisingly, we actually saw P53 in every single one of our KRAS cholangiocarcinomas. So P53 and KRAS seem to have a really strong synergy in terms of forming cholangiocarcinoma in this model. But on top of this, we identified a number of additional novel drivers that aren't necessarily recognized in cholangiocarcinoma as being key drivers, but were able to interact with KRAS and P53 in this context to induce tumor formation. So we focused kind of from this point on NF2 as a gene that seemed to have a lot of interaction with KRAS. Basically, we looked at principal component analysis of transcriptional data, and this suggested that NF2 with P53 and KRAS modified the transcriptional profile of these tumors significantly. So we're interested in how NF2 interacts with KRAS G12D to change the phenotype of these tumors. When we then focus just on NF2 and P53 guides, so we're not doing the library anymore, we're just losing NF2 and P53, we find that P53 and NF2 loss together with KRAS G12D results in much more fatal or worse survival in mice with these injections than just P53 loss alone or NF2 loss alone, or with KRAS. So I guess what this suggests is that NF2, P53 together synergize to exacerbate KRAS-driven tumors in this model.
Ed, can I interrupt you there for 2 seconds? So I had a couple of questions. If you go into the previous slide, we can see that you have modeled both G12V and G12D. Have you noticed if there are the same co-occurring alterations that are necessary to develop the tumor?
So we did NRAS G12V and KRAS G12D with that kind of question in mind originally. What are the differences? And there were a number of drivers that did come up uniquely in both the NRAS and the KRAS experiment, but it's not something we really followed up on, to be honest. I think there's a lot of further work to be done on the back of this study, and that's one of the main questions that we haven't yet answered. I can't remember off the top of my head, I'm afraid, what those specific drivers were, but we did see a small difference in some of the drivers that came up between these two groups.
Yeah, I thought it was just interesting considering the data that Tom just presented about the aggressiveness of the G12V and see if they are a little bit more autonomous on their own. And the other question was about the when you include the 3 different molecular alterations, are they simultaneously introduced into the mice, or do you have any way to analyze the sequential temporal order?
So the hepatocytes only take up the plasmids in response to the stress of the injection. So at least the genetic material, the plasmids that are being introduced, are all being introduced simultaneously. And I see no reason after that why the Cas9 edits or the genetic edits as a result of these plasmids would have any difference in delay or latency. So the presumption is all of these edits are getting done at essentially the same time, right at the start of this process, which maybe doesn't represent human disease too well, but in this model, that's unfortunately the way it works.
Okay.
Good question from me. Have we seen this, let's call it a potentiation of G12V with NF2 in other diseases at all?
Oh God, you're testing my literature knowledge here. There was, there is some papers in obviously other cancers, and I can't remember specifically what, which suggests that NF2 can, as you say, potentiate KRAS activity. I can't remember the specifics of the study, but I think there is some loose evidence in other models that there is an interaction between the two.
Thank you.
But yes, apologies, I haven't got a full answer for you there. It's definitely not something that's very well characterized, but there is small amounts of literature out there.
Thanks.
Okay. So we think that NF2 and P53 together can exacerbate KRAS-driven tumors. So we wanted to kind of pick this apart transcriptionally and try and find out what the differences are phenotypically between these double kind of loss and single loss KRAS tumors. So we looked at transcriptional upregulation between P53 NF2 KRAS tumors versus KRAS NF2, and also between the triple mutant and the P53 KRAS. So we basically looked at where NF2 and P53 are more than the sum of their parts. So where the co-loss of P53 and NF2 drives upregulation or downregulation of the same things compared to just a single mutation. What we found in terms of upregulated and downregulated genes was changes in a lot of protocoderins, but also a lot of Wnt signaling pathway components, both Wnt inhibitors and Wnt ligands as well. So this kind of in itself was slightly intriguing. And when we looked at the protein level, we looked at reverse phase protein array to look at a number of phosphorylation events in these 3 models. And what we saw is that GSK3 beta phosphorylation is increased. So inhibitory phosphorylation is increased by NF2 and also in the NF2 p53 group. But more interestingly, beta-catenin inhibitory phosphorylation and AKT activating phosphorylation are changed with NF2 and then further decreased or increased when p53 is there. So we see activation of beta-catenin, Wnt, and AKT pathways where p53 and NF2 increase these pathways further than single mutations alone. So we've got this collaboration of these mutations in the context of KRAS that drives these pathways. One of the main reasons this is interesting is because these changes make these KRAS tumors seemingly susceptible to Wnt and PI3 kinase inhibition. So when we treat these mice with either vehicle Wnt inhibitor, porcupine inhibitor, LGK974, or the PI3 kinase inhibitor, piclitosib, I think. Excuse me if I'm wrong. So we don't see a significant change between vehicle and Wnt inhibitor or vehicle and PI3 kinase inhibitor, but when we use a combination treatment, we see a significant increase in survival of these animals. So in the context of KRAS mutations, these extra mutations, including NF2, which is considered a rarer mutation, drive susceptibility to Wnt and PI3 kinase pathway inhibition. And this means that even in a disease where Wnt mutations, for example, are extremely rare, the co-mutations and the rarer mutations can sensitize or make cancers dependent on Wnt signaling as a pathway. We think this is a really interesting concept. And I just want to kind of punctuate that point by showing a little bit of human data. This is just transcriptional data from a study of human CCA, where we looked at metagenes for the Wnt beta-catenin pathway and AKT activation. And this is just to make a broad point that these pathways can be activated and seem to correlate to some extent in human CCA, in the absence of a large number of obvious mutations to the Wnt pathway. So what we're suspecting here, or what we're suggesting in a broad context, is that the less frequent mutations can interact with more common mutations to make these patients' tumors dependent on alternative signaling pathways such as Wnt AKT. And thinking about the wider mutational context as well as the transcriptional context and pathway activation within these tumors might open up not only new avenues for therapeutic treatment, but importantly might help us understand where some of the KRAS targeting maybe doesn't work, or why some patients are refractory to those sort of inhibitors. So yeah, just to conclude my last slide, really the point I want to
quickly go back to the previous slide. Sorry, just before you get onto your conclusion. Do you think that the upregulation of Wnt dependency, as it were, is dependent on the KRAS G12D? Do non-KRAS G12C driven cancers also rely on that pathway?
I'm sure there are instances where non-KRAS mutant cancers depend on Wnt signaling. So it's not necessarily a consequence of KRAS, but it's a way in which a KRAS driven tumor has been modulated. So yeah, I think there'll be other mutational profiles that are dependent on this pathway, but I think there'll be subsets of KRAS mutant tumors that are driven to susceptibility of this pathway through alternative mutations. So the Wnt activation isn't necessarily a KRAS thing, but it shows that KRAS signaling can be modulated, if that makes sense. So it's not kind of a blanket statement.
Can I ask you something, Head? Because I mean, your team has shown that the activation of Wnt pathway in cholangio more frequently comes from the immune cells, and we have seen from Tom's slide that actually KRAS inhibitors can rearrange the immune environment, and maybe that is what drives the response. So have you had any chance to characterize the immune microenvironment of these tumors and see what the presence of these other molecular alterations can change in the immune response?
No, it's a really interesting point. Short answer, no, we haven't looked at that. And I think there's a plethora of really interesting things that we can look up in following up this study. So I haven't got a clear answer for that, but I think it definitely is worth looking into whether some of the changes in Wnt activation, etc., are driven by microenvironmental interactions, and it could very well be the case. It's, as far as I'm aware, just not something we've looked into yet.
Okay.
The Wnt pathway activation in the NRAS one as well, or is it KRAS that seems to do it?
So we only looked, we followed up NF2, and we only followed up in the context of KRAS G12D. So the short answer is we didn't really look into NRAS. We had initially set up the study to compare and contrast NRAS and KRAS, and then we kind of found that these looking at these further interactions with things like NF2 was more interesting and more what we wanted to do. And so, long story short, unfortunately, we didn't look at these processes in the NRAS at all. It's one of those projects, unfortunately, where there's so many interesting things, but the PhD students kind of running them move on to bigger and better things, and the project kind of stagnates. So I might try and push Luke and see if we can kind of get the ball rolling on some of these questions, because I think there's a lot more to explore here.
Yeah, I asked just because I think, you know, the colon cancer story for KRAS G12C sort of, you know, followed the BRAF V600E story, where, you know, you got EGFR signaling upregulation and you had to combine it with an EGFR inhibitor really to see the efficacy. And I think it'd be really fascinating if that seemed to be the same thing that emerged in cholangiocarcinoma with Wnt being the driver, again, even without molecular alterations, if it really was a pathway that seemed to drive a lot of resistance.
Yeah, very interesting. Yeah, and I think kind of these models offer a lot of, you know, they're not perfect, but some of these questions can be answered in a quite a nice way using these sort of in vivo screens. So I do think we need to revisit some of these more specific questions in our lab and see if we can pick it apart using this library screen approach, really. But unfortunately, it's not been done yet.
There's just a couple of questions on the Q&A.
Sure.
One is just for clarification from Professor John Bridgewater. Sorry to repeat, but are you able to inhibit RAS-driven cancers by inhibiting, for example, Wnt?
Yeah, we've looked at this in not just KRAS-driven cancers, but KRAS Nf2 p53 cancers. So in that context, the context where we show that Wnt is upregulated, those tumors are, or survival at least, is improved by inhibiting Wnt, or inhibiting Wnt and PI3 kinase at the same time. We haven't looked at Wnt inhibition in pure kind of KRAS, or it has to do with something else. So KRAS P53 tumors, say, we haven't looked at that. But our suspicion is that you'd need one of these kind of extra rarer mutations to be driving Wnt activation and Wnt dependency for that to be effective.
Thank you.
And was there another question on the chat, or is that everything?
I think probably for the general discussion. So please.
Cool. Okay. Yeah. So I mean, just to kind of summarize the points I was trying to make, really, which is just that the mutational heterogeneity in CCA is going to, from my point of view, interesting, but also clinically an issue, I guess. And there are broader or rarer mutations that seem to interact with KRAS G12D. We can model these in our mouse models, and we suggest that actually certain rarer mutations can activate pathways with no clear mutation for that pathway. So Wnt being our example in this context, that you can activate things like Wnt pathway without Wnt mutations. And so in a patient context, understanding the frequency of these rarer mutations and whether there's a way to stratify these into specific drivers of pathways, you know, might help us inform targeting of these common mutations. So, and understanding when patients don't respond to things like KRAS G12D inhibition. Cool. And that's me. So thank you very much for giving me the opportunity.
Thank you very much, Ed, and thank you very much, Tom. So can we just encourage everyone to add your questions into the Q&A session, and then we will be happy to pose them to our speakers. So we have one that I think is for Ed, and Soravis is asking if you can briefly talk us through how you define the signaling pathway that can mediate the resistance. I think you look at transcriptomic changes, or have you combined with other analysis?
In regards to the upregulation of Wnt in these NF2. So, I mean, one of the ways that we've analyzed them transcriptomically, and we've looked at broad kind of metagenes for Wnt and AKT signaling, and they're increased. And also we've looked in these tumors when NF2 and P53 are lost, all in the context of KRAS, obviously. We've looked at those in terms of these reverse protein phase arrays and shown that the activation of kinase is important for Wnt signaling, and PI3 kinase signaling are inhibited or induced. So we've shown at the protein level and the transcriptomic level, in this specific example, that NF2 and P53 drive those pathways. We haven't shown that they induce any form of resistance to KRAS inhibition. We haven't looked at targeting KRAS in that context. But there's something we could probably look at, to be fair.
Yeah, probably maybe interesting also to see how much of this data would be translatable into humans, right? I mean, Tom has shown us that we have molecularly characterized from a genetic perspective human tumors, but we haven't complete all multiomics analysis yet. So probably that may be useful. I think we've also come to the end of the session. So if there are any more questions, please put them on the question and answer now. Juan, do you have any further comments or questions for our speakers?
Just one final question to Tom, really. Given what you've heard from Ed's presentation, what are your thoughts about where we should be developing this approach in the clinic? What do you need to know before we went down to studies that are combining Wnt inhibition?
I mean, I think, a little more preclinical modeling, probably with different, KRAS mutations and potentially other, mutations like NRAS, to see, again, is it like colorectal cancer, where it seems like anything in the MAP kinase pathway activates Wnt like it does EGFR in colorectal cancer, or is it KRAS mutation specific? I mean, the challenge is there aren't, currently approved Wnt inhibitors. So doing these studies, I think we aren't going to capture this in the emerging data from these studies that are going to look at NGS and other stuff. As Ed said, these aren't mutations that drive it. And so if you're not looking for it, we won't really find this from the existing studies. So I think, a little bit more preclinical modeling, but then really, I think a cholangiocarcinoma specific study that looks at it in combination, if it really is specific to this disease, that we see activation as a resistance mechanism of Wnt.
Thanks, Tom.
And I think that it was very interesting what you proposed, the data that you've shown, Tom, about the combination with immunotherapy. So for other targeted therapies, we have seen that the enhancement of the effect of immunotherapy comes from the fact that if you think, for example, IDH1 mutation, the metabolite can really change the way the lymphocyte can respond and can behave. So do you have any data to support why we have seen that synergy in the association between immunotherapy and KRAS inhibitor in the mouse models you presented? Any mechanisms that can justify that?
I mean, I think, Ed, I don't know if you were going to comment as well.
Right.
I think that some of the papers do go a little bit more into the mechanisms. We've seen a lot of this with these molecular therapies, that they do seem to have, broad implications in the immune system. Not all of them have panned out, like MEK plus PD-L1 hasn't really shown itself in most tumor types where it seemed to be effective preclinically, but I think absolutely worth following up on to see if it's real.
Thank you very much. I think we're coming to the end of our presentations. I'd really like to thank both of our excellent speakers for not just the good presentations, but allowing us to frequently interrupt and be able to just delve into some of the detail is hugely appreciated. Thank you both. And just to let all the attendees for the 29th of April. So please save the date for the 29th of April. The topic for discussion is going to be PSC-related cholangiocarcinoma, and I know there's lots of groups working on this. So we will certainly contact you with more details in due course about the next webinar. And in the meantime, we will be contacting you for some feedback and any ideas, suggestions that you have for topics that you'd like to delve into. Do please let us know. So with that, I'd like to thank my co-chair, Chiara. Thanks Eds and Tom again for a fantastic presentation, and thank you all for attending. Have a good rest of your day. Bye for now.
Thank you, everyone.
Thanks. Bye.
Bye. Thank you.